Hello friends, this video on molecular basis of inheritance part 2 is brought to you by examfear.com. No more fear from exam. Now let us talk about the structure of a nucleotide, whether it is RNA or a DNA. There are differences in their structure, but the concept of the structure remains the same. So any nucleotide, now we are talking about the structure of a nucleotide, and many such nucleotides join together to form a polynucleotide. Right? So first we will just understand the structure of one monomer unit. And what is that one monomer unit? That is a nucleotide. So any nucleotide will have three important components. And what are those components? Pentose sugar, nitrogenous base, and phosphate group. So these are the three important parts of a nucleotide. So here you can see in the figure, so this is the phosphate group. So here you can see the phosphate group. So this is the phosphate group. Then you have a pentose sugar. So where is the pentose sugar? So this entire structure which you see here, this entire structure is the pentose sugar. Why pentose sugar? Pentose, pent means 5. So here you can see this is a pentagon, 1, 2, 3, 4, 5. So it forms the shape of a pentagon. That is why it is called a pentose sugar. And then you have a nitrogenous base. So this is your nitrogenous base. Nitrogenous base means a base which contains nitrogen, or a nitrogen containing base. I'm sure by now all of you know what is a base and what is an acid. You would have studied all these things in your basic chemistry of class 9. So now what we have to understand is, first we have to understand each of these components in detail. What exactly is pentose sugar? How is it structured? How exactly are the nitrogenous bases, and how the phosphate group is? And then we will see how the three components are joined to each other. First, let us talk about the pentose sugar. So pentose sugar is anyways a saccharide, right? So pentose sugar here is a monosaccharide with 5 carbon atoms. That is why the name pentose. As I said, pent means 5. So you have a total of 5 carbon atoms. So where are those 5 carbon atoms? You see, this is 1. So carbon is not mentioned, so all these joints are carbon. 1, 2, 3, 4 and 5. So these are the 5 carbon atoms. Now this pentose sugar, there can be 2 types of pentose sugar. So if it is RNA, so in that case a ribose sugar is present. In case of DNA, deoxyribose sugar is present. So this is one of the major structural difference between RNA and DNA. So in RNA you have the ribose sugar. So here you can see this is ribose sugar, and in DNA you have the deoxyribose sugar. Now you might wonder, what is the difference between ribose and deoxyribose? So if you compare their structure, the only difference which you see here is in the OH group. So in deoxyribose sugar, you actually see that one of the OH group has been replaced with a hydrogen, and that is why it is called deoxyribose, because hydroxy group is removed, so it is deoxy. Right? So that's how you can remember. Anything D means decoloration, means the color has been removed. Right? So similarly, so deoxy, that means the hydroxy group has been removed. So that is how you can say here you have only one hydroxy group, but in this case of ribose sugar you have 2 hydroxy group. Now if you look at the structure of any nucleotide, how will you understand whether it will form RNA or DNA? By looking at the structure of the pentose sugar. So if the pentose sugar is ribose, it will form RNA, and if it is deoxyribose, then it will form DNA. Okay, now let us talk about the phosphate group. So what is the phosphate group? So here you can actually see the deoxyribose sugar has been derived from the ribose sugar, because if you see the structure is exactly the same, it's just that this oxygen has been removed. So the hydroxy group is replaced by this hydrogen. So the oxygen has been removed, and that is why the name deoxy. The next part is the phosphate group, and It is nothing but the inorganic salt of phosphorus. So you all know what is phosphorus. So it forms the backbone of polynucleotide chain along with the sugar. So this pentose sugar and the phosphate group, they together form the backbone of the structure of the polynucleotide chain. I mean, whether you talk about DNA or you talk about RNA, so the backbone is actually formed by the sugar and the phosphate. So when you talk about phosphate, how do we write phosphate? So normally this is how we write phosphate. So let us look at the third component, that is the nitrogenous base. So it is a nitrogen containing compound with properties of a base. So obviously it is base, so obviously it will have the properties of a base. Nitrogenous because it contains nitrogen. There are two types of nitrogenous base, and they are purine and pyrimidines. So these are the two types of nitrogenous base, purines and pyrimidines. So let us see what is a purine and what is a pyrimidine. So first we'll talk about purine. So it is a heterocyclic aromatic organic compound. So too many big terms. So let's understand it term by term. But however, I'm sure you would have studied about all these things in your organic chemistry. But still, just to keep things clear at this point. So what is heterocyclic? Hetero means different and cyclic means any cyclic compound, right, which starts from a point and then comes back and ends at the same point. So heterocyclic basically means a cyclic compound with atoms of at least two different elements. So when you have atoms from two different elements, you say it is heterocyclic. So how will you say that this is heterocyclic? Just look at the structure. So you see, you have nitrogen, you also have carbon. So if you look at the shape of the compound, it is cyclic, right? It is in the shape, I mean, it is somewhat circular. Wherever it starts, it comes back and ends there. Correct? At the same time, it has multiple elements. Like here, it has nitrogen, it also has carbon. So it is heterocyclic. What is the meaning of aromatic? Now, aromatic means a hydrocarbon with alternating double and single bond. So here if you see one double bond, then single bond, again double bond, single bond, double bond, single bond. So aromatic means alternate double and single bonds, alternate double and single bonds. So that is known as aromatic. So purine is a 9-membered ring. So what do we mean by 9-membered ring? So here if you see in purines, these are the examples of purine. Adenine and guanine are example of nitrogenous bases which are purines. Why? Because they have 9-membered ring. 1, 2, 3, 4, 5, 6, 7, 8, 9. Right? So if you look at both of these structures, they are 9-membered rings. So that means the points which you have on the ring or the elements which are present on the ring, that is equal to 9. And also the purines have a double ring structure. So this is one ring, this is another ring. Similarly here, this is one ring, this is another ring. So we have a double ring system. So here it has a double ring system. So here we can write it. So purines have a double ring system. What else? And it has 4 nitrogen. So if you see here, 1, 2, 3, 4. 4 nitrogen here, 1, 2, 3, 4. So here also you have 4 nitrogen and 5 carbons. So 4 nitrogen and 5 carbon. Right now we are only talking about the ring. So please do not count this nitrogen because this is outside the ring. So on the ring you just have 4 nitrogen and 5 carbons, and the rings are like double ring. The 2 rings joined back to each other. So that is why purines can be distinguished from pyrimidines by looking at their structure. So if it is a double ring structure and a 9-membered ring structure, then it is definitely going to be a purine. So examples of purine are adenine and guanine, as you can see in the figure. So these are the examples of purine. Let us talk about pyrimidines now. So these are also heterocyclic aromatic organic compounds. So they will also have alternate double and single bond. They will also have elements from at least 2, they will also have atoms from at least 2 different elements. For example, these are examples of pyrimidines: cytosine, thymine, and uracil. So here you see you do not have a double ring structure. So here you have single ring system. So how many membered ring is this? 1, 2, 3, 4, 5, 6. So 6 membered ring, as you can see here. So in this 6 membered ring, if you see that they also have alternate double and single bonds. So you see somewhere you have double bond, then again you have single bond. However, the pattern is little different from what you had in pyrimidines. So but again this is also cyclic, and this also has atoms from different elements. So it is heterocyclic and aromatic, but the ring is a single ring with six members. So here, how many nitrogen and carbons do you have? So in this case, we have 2 nitrogens and 4 carbons. So if you look at any of their structure, you will see there are 2 nitrogen and there are 1, 2, 3, 4 carbons on the ring. So looking at the type of ring present, we can distinguish whether a nitrogenous base is a purine or a pyrimidine. Now, examples of pyrimidine are cytosine, uracil, and thymine. So these are the important nitrogenous bases. Now, depending upon the structure of DNA or RNA, some of the specific nitrogenous bases are present. For example, uracil is an example of a nitrogenous base which is present only in RNA and not in DNA. Similarly, thymine is an example of a nitrogenous base which is present only in DNA and not in RNA. So these are the 3 important components as far as the structure of a nucleotide is concerned. Now the question is, how are these 3 components joined to each other? So in order to join any 2 parts chemically, what do we need? We need a bond. So let us talk about the bond which is present between the nitrogenous base, the sugar, and the phosphate. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.